like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong, in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong, in my happy place. And I'm right where I belong, in my happy place. And I'm right where I belong, in my happy place.
Thank you for joining us and welcome to the National University of Singapore Yong Lu Lin School of Medicine's Healthy Longevity webinar. I want to remind everyone tonight to use the Q&A function to ask questions. I'm always impressed by the number and quality of the questions, and I'm sure we'll have a lot of good ones tonight as well. We have Riekelt Houtkooper here tonight from Amsterdam, where apparently they're having a flood of biblical proportions from the rain. So he's dry and warm inside though, and we'll come back to him in a minute. But first I want to introduce Grace Kay, who's a research assistant in our Center for Healthy Longevity, and she'll be talking about blue light and flies.
Thank you for the kind introduction. Hi everyone. As mentioned, I'm Grace, a research assistant from Professor Kennedy's lab. Today, I'm presenting a paper from the Nature Partner Journals of Aging. Here, Song and his team have investigated the age-dependent effects of blue light exposure on lifespan, neurodegeneration, and mitochondrial physiology in fruit flies. Firstly, what is blue light? It is a major component of light-emitting devices, LEDs, such as your handphones, computers, and TVs. Unfortunately, our eyes are not effective in blocking it out. Hence, blue light can enter and damage the cells in the retina, located at the back of the eye. With the rising prevalence of blue light in this digital era, this raises safety concerns. Current research has focused on blue light damage to the eye. However, less is known of its effects on other cells. Recently, blue light was also found to reduce lifespan in worms. Building on that, the authors recognized that aging is accompanied by a decreased tolerance to environmental stresses. Coupled with the rise of blue light-enriched LEDs in homes and workspaces, they had two questions. Does constant blue light exposure throughout one's life negatively affect cellular functions? And does susceptibility to blue light change with age? To look at the effects of blue light on lifespan, they measured the survival rate of flies that were initially kept in the dark and then transferred to blue light at a progressively older age at 2, 20, 40, or 60 days old. They found that aging flies had reduced survival under constant blue light. The earlier the flies were exposed to blue light, the greater the reduction in lifespan. Moreover, as shown by the blue bars, young flies could survive in blue light for 2 to 3 times longer than old flies. So this shows that flies become more susceptible to blue light damage as they age. Similarly, lifespan reduction was observed in EYA flies, which are genetically edited to have no eyes. So this means that lifespan reduction is not solely due to eye damage. Now, the authors wondered if the increased susceptibility to blue light in older flies was due to neuronal damage. Hence, young and old flies were either exposed to blue light or kept in the dark for 10 days. Then, neuronal damage in the brains were measured. On the right are pictures of young and old fly brains. The red arrows pointing to tiny dots show the loss of brain cells, indicating neurodegeneration. We see that blue light exposure caused a significantly higher loss of brain cells only in old flies, but not young flies. The authors wondered whether these negative effects of blue light on lifespan and the brain were due to problems in mitochondrial function. Commonly referred to as the powerhouse of the cell, the mitochondria produces energy in the form of ATP to power different biological processes. Hence, the authors looked at ATP, or energy levels in the body. Energy levels showed a dramatic decline with age, even in flies not exposed to blue light. Additionally, blue light exposure moderately decreased energy levels, which was much more obvious in the young fly brains. In summary, other than damage to the eye, blue light exposure negatively affects the body, such as lifespan reduction and the loss of brain cells. Moreover, blue light is an environmental stressor that impacts organisms more severely with age. This was partly due to problems in mitochondrial function. Given that energy production already naturally declines with age, blue light exposure could worsen the age-related impairments in energy production. So, what do these findings mean? Before you turn off your computer screen in fear, I'd like to highlight that this study was done in flies. Hence, more research is still needed to confirm if these are also applicable to humans. In fact, I'm quite excited to see what these findings may bring in future. In addition to blue light blocking glasses, this could spur the creation of other inventions to limit blue light exposure. Thank you for your kind attention.
Thanks. Interesting finding. So looking forward to see some more research on that. We're going to keep a little bit on mitochondria, I think, today. I'm going to introduce Riekelt. He received his PhD in genetics of molecular metabolic diseases of the Amsterdam UMC-AMC, working on Barth syndrome, which is a rare mitochondrial disease. He then went to do a postdoctoral fellowship in Lausanne, where I was lucky enough to spend a month in Lausanne. It's a good place to be, and working on more common metabolic diseases. And since 2012, he's had his own group in Amsterdam, working both on inborn errors of metabolism, but also on aging. And so his talk tonight is on NAD as a target to promote healthy aging. And so welcome to the show, Riekelt.
Thanks, Brian. It's a great pleasure to be here, to be invited, and to present to you the work that we've been doing over the past decade or so on NAD and the direction we're taking this. So basically, in my group, the thing I'm mostly fascinated about is metabolism, which is shown here as this metabolic subway map, and the different influences that hit metabolism in several ways, and ultimately lead to what I call metabolic health in the context of rare diseases or more common diseases that also includes aging. And so we're interested in DNA, mutations, variants in the genome that can somehow tune this metabolic network, divert it from one pathway to the other, as well as nutritional factors as well as pharmaceutical factors, I should say, that also influence metabolism in different ways and tune this metabolic network to promote metabolic health or metabolic disease, and then we can reverse. So the topic for today's talk is on NAD, nicotinamide adenine dinucleotide, which a lot of people know from their biochemistry textbooks as a molecule that is involved in many different reactions in the cell. So it's involved in energy homeostasis, in glycolysis, TCA cycle, oxidative phosphorylation. But in recent years, it has become apparent, let's say the past decade or so, it has become apparent that NAD also is a cofactor for several enzyme families, including the poly-ADP ribose polymerases, the PARPs, the sirtuins, and also the CD38 enzyme, which use NAD as a substrate and produce nicotinamide. And in doing so, these PARPs, sirtuins, and CD38, they influence a wide variety of metabolic reactions that are all indirectly influenced by NAD. And so overall, NAD participates in hundreds and hundreds of enzymatic reactions and plays a role in basically all metabolic processes in the cell. And so maintaining homeostasis for NAD is, of course, of utmost interest for a cell to keep proper function. This plays a role in maintaining mitochondrial function, metabolic fluxes, redox reactions, but also inflammation, immunology, DNA repair, signaling pathways, All these things rely on proper NAD homeostasis for maintaining health. So given that NAD takes such a central position in these various pathways, there needs to be a proper homeostasis, proper balance of NAD. So, and the balance of NAD is maintained by, on the one hand, synthesis of NAD, and on the other hand, the consumption of NAD. And so in a very simplified schematic, this is basically how it looks. So NAD, if we take NAD for the time being as a central metabolite, will be consumed by several different enzyme classes, CD38, the PARPs, and the sirtuins, as I mentioned, each with their own functions. And on the other hand, NAD is being synthesized from various precursors. We'll get back to that in a couple of slides, including tryptophan as an amino acid, but also nicotinamide, nicotinic acid, niacin, which is a vitamin B3. And also some precursors that, or NAD boosters that have been more recently uncovered, like nicotinamide riboside, nicotinamide mononucleotide, and also the reduced equivalents of these molecules. We'll come back to that in a minute. And then there's also enzymatic pathways or signaling pathways that also lead up to enhanced synthesis of NAD, for instance, through AMPK. So this is the rough schematic that we'll be talking about also in the remainder of the talk. On the one hand, molecules that consume NAD and thereby deplete the levels of NAD, and also on the other hand, molecules that synthesize NAD or enhance the synthesis of NAD and thereby also maintain homeostasis. So it's evident that for a molecule that is so central to so many processes, that unbalanced NAD homeostasis can lead to diseases, human diseases. And there's, for this talk, I discriminate basically two classes of diseases. First of all, the primary NAD deficiencies. So these are typically genetically determined. So there are mutations in genes that are somehow involved in NAD synthesis. So that's, for instance, Hartnup disease, which is a disease involved in amino acid transport. Some of these enzymes are involved in NAD synthesis directly or in NAD resynthesis, but also in detoxification, for instance, of metabolites that are made from NAD. So NAD is converted to some toxic metabolites, and there's this detoxification system that basically recovers it. So this is the primary genetic deficiencies. We'll not talk so much about it anymore in the rest of the talk, because we'll focus mostly on the secondary NAD deficiencies. And the one that is most familiar to most people is probably pellagra, which is a classical disease that, well, I'm not a clinician, but I don't think this is a very common disease anymore anywhere in the world. It's basically a nutritional deficiency that, for instance, can be caused by excessive eating of corn, which is devoid of vitamin B3, and therefore there's a vitamin B3 deficiency, which leads to a very serious disease. There's also other diseases that are maybe not primarily caused by changes in NAD synthesis or NAD utilization. For instance, the glutamine synthetase deficiency, which has a secondary effect on NAD levels. Also mitochondrial myopathies, that maybe the primary deficiency is not in the NAD system, but as a secondary sort of adaptation, there is indeed changes in NAD homeostasis occurring in these diseases. And the final one, which of course is the topic for today, is aging and age-related diseases. I'll talk more about aging and age-related diseases. If you're interested in any of the other aspects, we wrote a review last year, which I think covers all of these various NAD-related diseases quite nicely. So for aging, we got interested in this about 10 years ago when we started looking into metabolic changes in preclinical models of aging. And we did that in mice. So we compared young mice and old mice. So this is mice that are 3 to 6 months old versus mice that are 2 years old. And we also did a similar experiment in C. elegans, in worms, and we found very similar changes. So very similar in both cases. We saw that there is a marked reduction in NAD in whatever tissue you looked at. So liver and muscle, those are the two main focuses for the work that we did. And we saw a marked decrease in NAD in these mice. And this was associated with an increased activity of PARPs, poly-ADP ribose polymerases. You can see here the PARylation, so PARP activity. And indeed, in old mice, we see that is massively activated. And considering that PARPs consume NAD, this may explain that the decrease of NAD in these older mice compared to the young ones. But the question was, how is this in humans? And that has been a long-standing question, and only recently we've been able to answer that. And this is through a collaboration with Lotte Grevendonck, Joris Hoeks, and Patrick Schrouwen in Maastricht University Medical Center, in a study that on our side was led by George Janssens, an assistant professor in my team, where basically the Maastricht group collected muscle biopsies and blood samples from a cohort of human participants that were classified as young. So these are typically student age, between 20 and 30, and also a group of older adults, which is between 65 and 80 years old. And these older adults were then subclassified as either trained individuals, so these were typically taking 13,000 steps a day. That's quite active, I would say. What we call normal elderly, whatever you think is normal, but these people took about 10,000 steps a day, which I think for an older person is still quite a lot. And then there was a group of pre-frail elderly. So these were functionally impaired and took about 6,000 steps a day. So from these individuals, we were able to obtain muscle biopsies. We performed metabolomics to identify if the changes that we found in previous years in mice were also found in human muscle biopsies. And so this is an overview of the various metabolic changes that we see. So on the left is a volcano plot where we see on the upregulated or the downregulated metabolites in older adults compared to the young. And for this comparison, we basically grouped all the older adults together. So no separation by health status, by activity level. And what we can see besides many other changes, but one of the top changes that we identified in older adults compared to younger is that NAD is lower in the older adults compared to the young, signified here by the blue color, compared to the red in the young. Interestingly, if we then subset by activity level, so comparing the young individuals and then the athletic, the normal, and the impaired older adults, we see that there is actually a stepwise decline of NAD levels with health. So people who are older but maintain a quite high activity level, they also manage to maintain their NAD levels, or at least they have higher NAD levels compared to an older adult who is maybe slightly less physically active. And even worse in people who are pre-frail or much less active compared to the others. So suggesting really that the findings that we had in in C. elegans and in mice are also conserved in human muscle. So the question was, can we fix this change? You know, if we consider this decline in NAD an unwanted effect, can we then fix can we replete NAD levels and fix the problem? And so for that, we need to understand a little bit the biochemistry of NAD, and this is the pathway that we typically look at. There's a lot of details here. If you're interested, again, the paper is published, so you can look it up in more detail. But there are several ways to increase NAD levels. So one is a primary synthesis pathway from the amino acid tryptophan. The other ones are nicotinic acid, which is outside my screen somehow, but also the ones that are more recently have received a lot more interest is nicotinamide riboside, nicotinamide mononucleotide, and some of these reduced precursors. Each and every one of them, after supplementation, will be converted to NAD in a multi-step process. And there's quite some evidence from preclinical models that these interventions work. So nicotinamide riboside is very selective, first of all, I have to say. There's many, many more studies. I picked a few that we were closely involved with. So this is a study that we did with nicotinamide riboside in the context of obesity and diabetes. And we see that if we feed mice a high-fat diet, that they're actually protected against body weight gain. We saw that nicotinamide riboside extends the lifespan of the C. elegans. It also extends the lifespan in mice. And also this is a sort of a neurodegenerative phenotype in worms. where the mobility of the worms was assessed after a couple of days. And what you can appreciate is that if there is a vehicle being given, these C. elegans, they move much less compared to C. elegans that receive nicotinamide riboside. So there's extensive evidence, I think, from preclinical models that nicotinamide riboside, but also some of the other precursors, have a beneficial effect on aging. So that's summarized a little bit here. So there's many effects from many different studies. There's an improvement of fertility, there's an improvement of inflammation, there's an improvement of cardiac function, and all these other things as well. Of course, the question was, how does this translate again to humans? Because in the end, that's what we want to do. So this is a study we've been involved in. There are several other studies have been published in the meantime as well. But this was a clinical trial together with the Maastricht team again from Patrick Schrouwen, in which we treated individuals with nicotinamide riboside for a period of 4 weeks or 6 weeks. And tested, in a crossover model, tested the effect of the intervention of nicotinamide riboside compared to the placebo. And so we took muscle biopsies from all these individuals and assessed if NAD was actually changed. And strikingly, on the right side, we see that in muscle samples of individuals receiving nicotinamide riboside, we don't see any increase in NAD that we would typically see in quite a few of the tissues of the mice. On the other hand, we do metabolomics, so we don't measure just NAD, we measure also all these other intermediates of the same pathway. And what we do see in the muscle is that several of the intermediates like NAAD and also methylnicotinamide are increased. And this to us suggests that there is flux going through the pathway. So probably the nicotinamide riboside is going in, it's being converted, but we don't see a change in the steady state NAD. So ideally you would want to measure fluxes with maybe stable isotopes or something. But I think based on this, we can already say that there's something at least going on. It's taken up, it's being metabolized, and it's coming into the muscle. At the same time, we don't see so much of an effect on the clinical phenotypes. So for instance, these are all parameters of insulin sensitivity, whole body glucose homeostasis, and we don't see any significant changes whatsoever. We do see some minor changes in acylcarnitins, for instance, in the muscle that could be a reflection of a beneficial effect. We see some changes in sleeping metabolic rate that could also be suggestive of a beneficial effect. But overall, the effect is, well, not optimal yet. So the question is, why is the translation to humans so far limited? We've seen that actually quite a few of these precursors that have been used in the clinic for age-related diseases or have been tested for age-related diseases have limited translatability. So why is this? Is this the duration of the study? Is maybe 4 to 6 weeks not long enough? Should we treat longer? Is the doses maybe not high enough? Could be. I think another important factor is that a lot of these studies focus on relatively healthy individuals. Maybe they have some diabetes, some pre-diabetes, maybe they're overweight, but typically they're not very, very sick. So that could be a factor as well. And the other part that we wanted to focus on is to identify molecules that maybe have a more pronounced effect on the NAD intracellular pool. And so this is the work that a talented postdoc in my group, Ruben Zapata Perez, who now leads his own group in Spain, came up with in collaboration also with his PhD supervisor, Alvaro Sanchez Ferrer. And basically the idea that they had is to make new precursors for NAD, new precursors for NAD based on a pyrophosphatase enzyme from E. coli that would be combined or would be fed NADH as a precursor molecule. And this pyrophosphatate then makes NMNH, so the reduced version of nicotinamide mononucleotide from NAD. And so the stuff that Alvaro and Ruben did is really to develop this procedure and develop the purification procedures for NMNH, which is really a complicated procedure. And I hope you're not going to ask me questions about that. They managed in the end to identify the purification procedure to get pure NMNH. So the question was, can we use this NMNH, this new molecule, as a new NAD precursor or NAD booster. And so these are some of the first experiments that we did, exposing different cell cultures to NMNH and comparing always to NMN, which is the more classical NAD precursor, NMN in the oxidized form. So we tested in AML12 cells, which is hepatocyte cell line. We tested a brown adipocyte cell line, again, a liver cell line, fibroblasts, neuroblastoma, and a HeLa cell. And in each of these cases, we see that NMN, as expected, and this was an increase that we were excited about a couple of years ago. This is an increase of NAD after supplementation with NMN. But then if we give NMNH, our new molecule, all of a sudden, this change after NMN seems like nothing. All of a sudden, there's a tenfold increase or even more of NAD levels after supplementation with this NAD booster. Of course, we wanted to test also if this was true in mice or in an intact mammalian model. So we chose mice and we injected mice with either NMN, 250 milligram per kilogram, or NMNH. And after several time points, we harvested blood samples and took the tissues. So this was only basically 2 injections of NMNH, and this was not a longer treatment duration. So initially what we see here after injection of NMNH compared to NMN is that with NMN, we see an increase in NAD levels in the blood. This is in blood after 1 hour. After 4 hours, this is already lower. And then it's still lower after 24 hours. But with NMNH, we see after 1 hour, we see a massive increase in blood NAD. So this is whole blood. And this is sustained at least for these 4 hours. And also the next day, after injection, repeated injection, we still see that there is a much higher NAD levels compared to the vehicle control. So NMNH indeed also in mice, when we supplement them by injection with NMNH, we see a massive increase in NAD. And this is after 24 hours, we harvested the tissues. We took the liver, we took muscle samples, adipose tissue, brain, and all these things. And also in these tissues, we see that NMNH is much more potent than NMN in increasing the NAD levels in tissues. And this is especially true for liver and kidney, I would say, but also in other tissues where maybe NMN is not able really to increase the NAD levels. So for instance, here gastrocnemius muscle, we do still see that that NMNH has the capacity of increasing NAD levels even in these more difficult tissues. So to conclude my story, we've seen that the decline of NAD is observed across organisms, including humans, during aging. Interestingly, when in our human clinical trial, we found that this is also associated with health status. So people who are more active seem to preserve their NAD levels better than people who are less active. So this could be an interesting avenue to pursue also, and to understand really the dynamics of NAD under these different health conditions. The classical, if I can call them that, the classical NAD precursors, they show limited translation to humans so far. I think more studies should be done in different cohorts. I think also there is an opportunity there for maybe studying, like for instance, rare genetic diseases where mitochondrial function is impaired. Maybe there the NAD precursors or NAD boosters have a higher potential. But at the same time, also there's a potential for these new precursors. They're difficult to work with because we cannot get them in large quantities yet. But they are very effective in raising NAD levels in both blood and tissues. And so it could be attractive to optimize this further and work towards clinical application. As I said, it's too early for that. We don't know much about what it's really doing and whether such high NAD levels are actually going to be good or not. So I'd be very curious how this will develop. So with that, I will close the lecture. I would like to thank the people that were mainly involved in the work on our side. That's especially Ruben Zapata Perez and George Janssens, but also the rest of the team. Also in our pathology lab, where we did quite a few of the studies related to NMNH. Our collaborators on the NMNH work, especially from the University of Murcia, and also our colleagues in Maastricht University, where we collaborate a lot on the clinical studies where they take most of the time the muscle biopsies that we study, and also our colleagues in Lausanne, Carlos Canto, with whom we collaborate, also on the NMNH and the NRH, and of course the funding agencies that supported this work. And before I give the word back to the chair of the meeting, I would like to highlight this. We've recently been awarded a European grant to study NAD. It's called the NAD International Training Network. And so what we'll be doing is to host 10 PhD students across Europe with different partners, exposing them to both academic and industry work related to NAD. And we're currently recruiting, on my side, we're recruiting 2 positions. Those are the QR codes here. But also at the same time in University of Copenhagen, we have positions open. Also in Finland, we have a position open, and the others will follow soon. So if you're interested, or if you know someone who might be interested, please feel free to share this with them. And with that, I'll close, and I'd be happy to take any questions.
Thanks. It's a very exciting talk. I mean, I'm still getting over the fact that normal people over 65, and this is in Maastricht, these people are taking 10,000 steps, and the impaired people are taking 6,000 steps. I don't even think the normal people over 65 in the US take 6,000 steps. So these people are drinking Red Bull, or where'd you find these people?
I've been surprised about this as well, and we had quite some discussions about calling this the normal aging group, because 10,000 steps is for me also. I mean, I don't take 10,000 steps, and I'm not 65 yet. So,
yeah, it's impressive. So I wanted to ask a couple technical questions to start. You know, there's been a lot of debate about how to measure NAD levels. So how are you doing that, and what are your thoughts on the best ways to? I mean, do you need to do just NAD or NADH ratios, or what's the current thinking on this?
Yeah, so for me, I mean, we typically use 2 techniques to analyze NAD specifically. So we do enzymatic cycling assay, which is a classical biochemistry assay to measure NAD levels. And you can take NADH in a separate extraction. You can take it along if you're interested in that. At the same time, we developed this technique with metabolomics to measure NAD, NADH, and also several of the other precursors. Or intermediates, and that's working very well. It's actually very reliable. We've validated that method extensively, and we use a stable isotope NAD to really quantify NAD levels under these conditions. So that's typically how we do it. And with respect to just measuring NAD or the ratio, I think the ratio can be interesting, but I think the absolute levels are certainly also very valuable. Because sometimes you just see one moving and not the other. But I think to get the full understanding of what's going on, also, for instance, when in relation to these reduced precursors, initially we thought that maybe the reduced, so the NADH would go up primarily and not so much NAD. But actually the reverse is true. NAD actually goes up more than NADH. And so that's also an area of, I think, investigation that we need to go to understand how this oxidation process occurs under these conditions.
Yeah, I was going to come to that, but since we're on that, I was trying to, maybe you said this and I missed it. I was trying to understand the rationale for why the reduced form of these molecules would be more effective at driving NADH level, or sorry, NAD levels.
Yeah, so the main thing I can say there is that it uses different enzymes to produce NAD. And the main thing is that rather than nicotinamide riboside kinase, NRK, which is used for NR, to convert NR to NAD, is the adenosine kinase that is actually used for the reduced precursors. And that's true for both NRH and for NMNH, the one that we studied. So it could relate to this, just different enzymes that are involved. Other than that, the pathway is pretty much similar. It's the same transporter, transporting it into the cell. There could be something also in relation to subcellular distribution that we don't understand yet, but for sure we know that adenosine kinase is the one that phosphorylates.
I mean, that could be an explanation. And so the experiments you showed were through injection, right?
Yes.
So have you tried oral administration?
Yeah, we've tried oral. We were afraid, based on just the biochemistry of the molecule, that it would be acid labile. And so that's why we chose initially for the injection strategy to avoid basically neutralization inactivation in the stomach. And that worked very well. Then we did also a study where we gave it orally and we don't really see an effect. So it seems indeed it's unstable. I think what would be attractive there is to see if you can package it in some way to make it past the intestine and then get released. And then maybe you get the effect. But that's an experiment that we haven't done yet.
With our studies with AKG, we use a sustained release version of it to get it past the gut. So I think that might be a promising approach. It seems to work in that case.
That would be very attractive to explore that, actually. So
yeah, I say that partly because there's some similarities between AKG and NAD. I mean, these are highly used molecules. There are high levels of them. They participate in many chemical reactions in the cell, and that kind of brought me to the question with NAD is that, and I think you showed that very nicely with the metabolomic studies, is that you might not expect NAD levels to go up that much. I mean, the body is very good at using NAD. It's more it's the reactions that NAD drives and the metabolic changes that it stimulates that might be a better measure of whether any particular booster is working or not. And so, the body is really good at using this molecule. So it's not like a drug where you might expect it to accumulate in the body. So I think that using the metabolomics is a very effective way to get a better feel of whether you're doing anything or not.
Yeah, I think the next step will be the fluxes. And I mean there's people working on NAD fluxes, whole body fluxes already. And I think that's really the way to go. It's complicated and it's very expensive. So not everyone can do it. It's also the reason why we are not immediately jumping onto this. But I think it is a very important way of showing really that these precursors or boosters are actually being used and reach the tissues that you're interested in. The other thing is that also in preclinical models, we've seen that the muscle is not the best tissue to look at. Typically, we see great effects in the liver and the kidney. The muscle is always so-so. So even for NMNH, our new favorite molecule, we do see that it increases NAD levels in the muscle, but it's not very striking. So I think, focusing on diseases that maybe affect the liver or the kidney would be also an attractive way forward.
It sounds like you're taking a disease-oriented approach to try to test these molecules in the clinical studies. Can you say more about that?
Yeah, well, I mean, we're not so much into the clinic yet, at least not with age-related diseases. We're focusing currently on mitochondrial disease, so rare mitochondrial diseases. So it seems that we recently did a study in fruit flies with Barr's syndrome, and Barr's syndrome, as you mentioned in the introduction, is the topic of my PhD. And this was a collaboration with the University of Detroit. And they found that if they exposed fruit flies with Barr's syndrome to nicotinamide riboside, that these flies don't exhibit their exercise intolerance anymore. So typically the exercise capacity is 50% less. And now if you give them NR, all of a sudden they exercise like normal. And so this is very promising because this is a vitamin that is very accessible, seems to be very safe, and a population of people who are suffering really from a very severe mitochondrial disease. So this, putting one and one together, we hope to launch something in that direction. It's a little bit too early to say, but that's,
Maybe you could expand a little bit on the links between NAD levels and different mitochondrial functions and exactly what's happening in the mitochondria when NAD goes down and when you hopefully bring it back up.
Yeah, so that's a great question. I mean, it depends a little bit on what happens. I mean, whether NADH also is going up, for instance, in some of these conditions, you really see the opposite of oxidative stress, so reductive stress, which is not necessarily better than oxidative stress. It's equally bad. So under these kind of conditions, especially mitochondrial diseases, you often see that the reoxidation or the oxidation of NADH to NAD plus is not working properly. For instance, because of primary deficiency of the respiratory chain. But also in the case of Barr's syndrome, where there's more a problem with the lipid bilayer in the mitochondria because of cardiolipin, mitochondrial specific lipid that is changed. And So yeah, there could be many different things. It also depends on where you look in the cell. It's not strictly the mitochondrial NAD NADH balance that is disrupted, but also this is transmitted to other parts of the cell.
I always find it amazing that we spend so much time on oxidative stress and don't think about reductive stress at all. And
yeah. No, exactly. I mean, and oxidative stress has received, of course, a lot of attention, but reductive stress, not so much. I mean, there's more and more people now getting interested in it, but it's very clear that it is important to consider as well.
Yeah, the name of the scientist is not in my head, unfortunately, but there's people studying reductive stress in the context of laminin-related progerias. And it looks like the data is quite interesting. Hopefully, I'm not, sharing things that shouldn't be shared. But it also awakened me to thinking about reductive stress as well.
Well, and that's also Vamsi Mutha, for instance. He's really studying mitochondrial diseases and also forcing basically reoxidation of NADH on the cells, which is really improving also the sort of preclinical benefit in these models.
You mentioned a little bit, you didn't come out strongly about this, but you mentioned that PARP may be one of the things that's depleting NAD. I know there's also arguments for CD38, doing that with aging. Is it both, or is one more important than the other, or what's causing NAD to go down?
Yeah, that's a great question, and it's probably I'm trying to evade that question a little bit because I simply don't know. I mean, we've seen in our models, because we looked at PARPs, because we had a good assay to look at the activity of PARPs with this antibody. We know that that's certainly going on, but we haven't done any of the mechanistic conclusive experiments to rule out whether it's one or the other. We have done some experiments in C. elegans where we used the PARP knockout. And then so that was actually increasing NAD also and extending lifespan. So PARP seems to be involved somehow, but I'm definitely not excluding CD38 because I think also the preclinical evidence, for instance, in knockout mice of CD38, that those data are also very interesting with respect to the development of metabolic diseases and like age-related phenotypes.
The PARP story is potentially a very elegant story, right? Because you can imagine you have more genome stress and more damage, which is inducing to extra physiologic levels as you get older, and that's depleting NAD. I'm always trying to think about ways to connect the dots between different aging pathways.
So that was indeed the simple thinking that we had also at the time of the paper, at least, that with age there would be more genotoxic stress, PARPs would become active, and because PARPs use massive amounts of NAD, this means that NAD was going down. And at the time, we were really, sirtuin-centric, I would say, like most of the field. And so we were looking at the activity of sirtuins by also measuring deacetylation of specific sirtuin targets. And we see indeed that they become deacetylated under these kind of conditions. But I think probably our view on the whole biology and the mechanism there was a little bit too simplistic at the time. I think it will probably be a little bit more complex than that. That simple route of genotoxic stress, PARPs, NAD decline, sirtuin is probably too simple.
Everything is always more complex. Did you happen to look at SIRT6? How much does NAD drive SIRT6? I ask this question all the time. The audience is probably getting tired of it, but do you have any thoughts on this?
No, we haven't looked at that actually. So I can't really say. I mean, SIRT6 obviously is an interesting one. It's a little bit different than most of them, and probably the best one that is really causally linked to aging phenotypes. So, but we haven't looked under these kind of conditions whether SIRT6 is active or more active. And also in our recent studies with these precursors, we haven't really looked anyway at sirtuins. Also with the NMNH, we haven't really looked at the acetylation of whatever targets of whichever sirtuin you can think of. So no, we haven't checked that.
I'm trying to get this in my head, right? You have NMN and NMNH, and you're adding the same amounts, I presume, in the in vivo studies. In one case, NAD is going up and down quickly. In the other case, it's going up and sustained. Does that mean there's most of the NMN is getting wasted or not used appropriately, and the NMNH is stored better and then converted over time? I mean, how do you imagine this happening?
That's a great question. I mean, I don't know the dynamics so well. I mean, because it's also super fast. It's not like there's a delayed response with NMNH. Actually, it's faster typically. So the NAD increase, we see it happening faster when we expose cells to NMNH when compared to NMN.
I mean, maybe it just gets into cells better.
Yeah, although I mean the transporter is the same. Of course, that doesn't exclude any difference in the dynamics, but it seems to be the same transporters. There could be other factors, of course, making it go into the bloodstream and then into tissues faster.
It seems to me like this is a more general question with aging, is that the interventions that we found, and you could put sirtuins, inflammation, NAD, AKG levels, all these things into this category, is that we're not doing something super physiologic. What we seem to be doing is restoring a more youthful metabolism or transcriptome or whatever we're doing. We're restoring the things that are getting lost with aging rather than doing things super physiologic. Do you think that's true? And it's just trying to combat the changes of aging and not make a new metabolism that has some altered effect on aging.
I tend to think that's the way we should go, at least, to restore basically to a youthful phenotype rather than super physiological. And for instance, I think with NMNH, I mean, this 30-fold increase that we saw in some cell lines and also in tissues, really a massive increase in NAD. I cannot predict right now, without any supporting data, what this will do to a cell. Whether at some point also this NAD becomes too high. I mean, initially we didn't expect that it could go this high, because with NMN and NR, just the oxidized, like the old versions, when we would treat mice or cells with NMN or NR, we would always reach a plateau after 250, 500 milligram per kilogram. But with NMNH and also with NRH, we're breaking that ceiling massively. So it seems that there is no inherent limitation for that ceiling, that apparent ceiling that we thought was there like 10 years ago. Maybe the ceiling is now what we reach with NMNH and NRH, but I don't know, still don't know, also because we haven't done long-term follow-up of the treatment, what this will do to organismal health.
Also,
so that's also why I'm a little bit cautious with these molecules. I mean, I wouldn't take it immediately, even if it was available, I wouldn't take it.
Well, maybe you can take it and come back on the show in a year, and we'll see whether it's a good idea.
I think I'll leave it to others first to explore that further, and I hope there's really companies making NMNH and NRH in larger quantities and for a cheaper price, so that we can actually do it and package it. The other thing.
Yeah. So, Aim, can you tell us what the questions are today?
Yeah, thank you, Brian, and thank you, Professor Houtkooper. It was a very interesting talk, but we have a lot of interesting questions. Many questions are repeated around one issue, and I guess you could kind of predict what issue that was about. But before we go on to that, let me ask a few fundamental questions from the audience. So the first one of our audience asked, why can't we directly just supplement NAD in our body? Why must we go to other molecules?
Yeah, so NAD will be broken down in the bloodstream, so it will not enter cells as NAD. So I think typically the studies, because there are studies that have been described with NAD supplementation of the whole molecule, and I have a hard time believing that actually what we're seeing is really NAD going in, entering the cell, and then intracellularly elevating NAD levels. I think what will happen if you do this, NAD will be broken down and then the individual pieces will be taken up.
Yeah.
So actually giving a precursor is more effective.
Do you think that supplementing NAD in any form impacts the body's intrinsic ability to produce NAD?
Well, it's certainly increasing NAD, but whether it's increasing the intrinsic ability to increase NAD, that's of course something else. As far as I know, from our own data at least, we don't see that, for instance, there's an increase in the expression of all the enzymes that synthesize NAD under these conditions. So I don't think necessarily, at least based on what we've seen, that we see an increased capacity to synthesize NAD. It's just a supplementation of the substrate that will lead to higher levels of NAD.
Actually, the question is about whether it's decreased, but we probably don't have information.
No, we haven't seen that, no.
Yeah, okay. So among other supplements, would you say that NMNH seems to be the most effective to increase NAD levels, even compared to NAM or NR?
Yeah, so clearly, at least in the preclinical models that we've tested, so cell lines and one-time injection in mice, and I think there's another study where they did a slightly longer treatment, but in all cases, the papers that have been published so far with NMNH and NRH, so the reduced precursors, they always show that they are much more effective in increasing NAD levels compared to what I call classical precursors, NMN and NR. So that's absolutely true. I think the evidence so far for NR and NMN in preclinical models is much more overwhelming. There's much more out there. So there's many, many more studies. So I trust that data. Well, I trust the other data as well, but I think there's much more evidence to support beneficial effects for NMN and NR.
All right, this is Brian. Do you want to weigh in on which is better, NR, NMN? We get asked that all the time.
Yeah, so for me, I think both are equally good. I know there's a fierce debate going on, mostly from manufacturers and inventors related to NMN and NR. But I think the evidence so far, and I think also the biochemistry behind it, suggests that both will be equally effective. Of course, for NMN to enter the cell, there's one more step, enzymatic step in between, because NMN first needs to be converted to NR before it enters the cell. But I don't think that will be a major limitation for sort of its use in the clinic, in day-to-day life. So for me, they're equal, and also the same is true for NRH and NMNH. So we happen to be studying NMNH because that was available to us, because that's what Dr. Zapata Perez made for us before he came to us. But there's also other studies, actually 2 papers that I'm aware of that tested NRH with equal, very similar results.
Maybe a tricky question extended from the previous one. Do you think the best molecule that the molecule that is the most effective in increasing NAD levels would be the most effective or most beneficial for health?
Yeah, that's a difficult question to answer. I think that's exactly what we need to study in more long-term follow-up experiments, because I don't think that's necessarily the case. It's true that in vitro and in these mice, NAD levels go up, but as I mentioned also in my response to Brian, I have some hesitation, whether increasing NAD levels 30-fold will be good or not. So I think we really need to understand more of what this high levels of NAD is going to do. If it turns out that these high levels are harmless or actually beneficial, then yes, then this molecule may be the more attractive way to go. I think for so far, also the clinical trial that we are considering will move with the more classical precursors. And I know some studies also, for instance, from Anu Suomalainen's lab, where they studied NAD precursors in the context of mitochondrial myopathy. They studied niacin, which is even a more prehistoric almost precursor. If I call the other ones classical, then this one is prehistoric. That's already decades old and a very safe way of medication. It just has a side effect, which is it's causing flushing, which is very uncomfortable, I was told. I've never tried it myself, but it's really annoying, but it's a very safe molecule. So if you have a very severe disease, then maybe this is an attractive way to go.
Okay, thank you. Now I'll go to the most popular question. There is a recent study, you might have seen it, from the group in Switzerland, published on Biosensors and Bioelectronics journal, and it talks about NR supplementation effect on cancer metastasis. So that seems to promote at least the spread of cancer in mouse models. I think you can spend a couple of minutes answering this one, because a lot of people put it up and they are quite concerned about this side effect.
Yeah. So I haven't read the paper. That's the first thing I should say. I followed the discussion. I saw the discussion online, and from what I understood, so one of my close collaborators, Carlos Canto, was also a co-author in the paper. I saw he commented on some of the things that were posted online. To me, what I understand from the discussion, but again, that's without reading the paper, so I should be cautious here. But from what I understand from the discussion is that it's really a premature study. So it's based on cell culture models, mostly in a very specific context. So I think it's really too early to say. I guess more and more thorough studies need to be done to really say if NR is causing metastasis or not. So that's probably the politically correct answer. And to be honest, I don't have another answer.
Yeah, I think apart from cell culture, they kind of supplemented in a few number of mice, and the mice are injected with breast cancer cell line, and then I think they just found more cancers in the ones that are supplemented with NAD. But again, yeah, we were having a debate in our lab.
That's another indeed good that you remind me, because I saw a comment on that as well, because it's a very small group of mice. I think it was only 9 mice or something. True. And yeah. I think in the one group they had 4 mice with cancer, and the other group they had 6. So I think it's simply too early to say. I think if these effects in a cell culture model were observed, I think it's still worth publishing. And basically leading up to more thorough studies in the future. I don't disqualify that research on the basis of what I've seen in the discussion. But certainly, I think that was also what Carlos mentioned in the discussion that I saw evolving, is that there's definitely more research needed to explore this.
And do you foresee any other side effect apart from this one? Of NAD or supplementation?
I don't think so. I mean, I don't foresee any major side effect right now. I mean, that's, of course, the long-term studies will need to be done, but I think so far it's proven to be quite safe. Of course, it's going to be different if you take this thing for 20 years, it's going to be different than if you take it in the context of a very specific disease, where maybe you need to take it for months or a year. That's a very different situation. So I think more safety studies need to be done. I think that's apparent. That's probably true for any molecule that you study in the context of geroprotection. So,
at least in the shorter term studies, I don't think there have been a lot of adverse events that have been reported, right?
No, they are considered really quite safe. And that's also what makes it attractive for us, for instance, in the context of this mitochondrial disease, to just move forward. And initially, I thought we would first go to mice, but actually, when I was at a patient day, and the patients just said, "Just give me the thing, we want to go ahead." So that's why, together with the clinical teams, we're currently discussing, how should such a clinical trial be set up, because that's a little bit more complicated than for a common disease where you can maybe find 10 or 20,000 people to participate. Here, we only have 200 patients worldwide.
One more question, maybe, Aim?
Okay, I'm not sure if I'm going to the first one. Okay, this is a short one, so maybe we can squeeze 2. Do you think there are ways to increase NAD levels through lifestyle modification instead of using supplements?
Yeah, it seems that lifestyle modifications do increase NAD levels, including fasting, as well as exercise. So those studies, at least in preclinical models, have been thoroughly investigated. We do see the association also in our study, where people who are physically more active do have higher NAD levels. But I think that's, especially in an aging community, something that still needs to be further studied.
Brian, do we have time for one more or not anymore?
I think we're just about out of time. Sorry, Aim. And,
okay, no worries.
Thank you for
so maybe we can invite you. Yeah, that's okay.
You can come back. Yeah. You should come back, because, I was going to ask you to detail all the purification steps for NMNH from E. coli, but we've run out of time. So maybe next time.
Yeah, that's a pity. Next time I'll start with that.
Sounds good. All right. Thanks a lot. I want to remind everyone to use the chat function of the panelists and all attendees option to leave your comments on the show. Look for news for the center and also the School of Medicine and the end credits. We're constantly looking for members who are driven and passionate about longevity research to join our team. So there are all kinds of career opportunities available if you're interested. You can go be a graduate student with Riekelt if you want, but you can also stay in Singapore. I'm sure there's plenty of room for everybody. We launched our Center for Healthy Clinic at Alexandra Hospital last September. With the conference, if you don't manage to catch it, you can find the recordings to those conference presentations online now. I want to remind you the next episode is December 1st with Uwe Sunde, and the title of his talk is Longevity and Patience. So I'm really interested to hear the connection there, not patients as in people in the hospital, but patience as in waiting for me to get through the end of the show. Also, I want to leave you with a video from an 80-year-old bodybuilder. Thanks for joining us.
I am Ernestine Shepherd, 80 years of age. I started training at age 71. I was named the oldest female competitive bodybuilder at the Guinness Book of World Records. I had never done any type of exercising before. I have been in 6 or 7 competitions. I win, but some I come out second and third, and I'm pleased with that. I'm up every morning at 2:30. After I have my devotions, I walk and I run, approximately 10 miles. I'm at the gym at 7:30, and I have a group of ladies that I train. Down. 1. I train all ages. One of the youngest, I think she's about 20 years of age. And the rest go from 40 all the way up to 80. When people come to my class for the very first time, they are really, really surprised. They said, I didn't think this exercise would be this tough, but if you can do it, we are going to do it too. I really don't feel as though I'm 80 years of age, but I am. I want to keep training until my day is done.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.